Welcome back. In this segment, we're talking about muscles and muscle contractions. With that, let's begin with drawing what a muscle actually looks like. So, to the document camera, we're going to draw a skeletal muscle. And from there, we're going to segue into talking about how these muscles contract, how they contract to move our bones to where we want to be, and our hands, our fingers, all of those things. So this is a blow-up of a skeletal muscle. Within this skeletal muscle, there are what are called fascicles, and these are groups of muscle fibers. So again, these are called fascicles. Within these fascicles, let's blow this up again. There are muscle fibers. And within each of these muscle fibers, there are, again, little groups of what are called myofibrils. Within the myofibrils, there are groups of what are called myofilaments. And in each of these circlet of myofilaments, there are two myofilaments called actin and myosin. Okay, so these actin and myosin myofilaments are what actually are going to be controlling the muscle contractions. So let's go ahead and draw out two of these thin filaments. So let's first draw an actin filament. So actin molecules are strung together and they wind around each other. So they kind of braid together here to form this two molecule filament structure. On these actin molecules, there is a compound that wraps around, and it's called tropomyosin. And attached to this tropomyosin, there are compound called troponin. Okay? And these troponin molecules attach to the tropomyosin filament that wraps around the two filaments of actin molecules. Okay? So each of these beads is an actin molecule. Okay? The myosin myofibril is made up of two myosin molecules that are wrapped together, and off the top of each of them is this little golf club-like head. And so there's two of them wrapped together, and they look something like this. And so this is myosin. So actin and myosin, the two myofilaments. And on the actin, this tropomyosin covers binding sites for these heads of the myosin. And as we go through talking about the muscle contraction and how that occurs, we'll see how these golf club shaped heads bind to actin molecules, and in particular on binding sites that are covered by this tropomyosin. So let's look at what these look like in the book. So we'll zoom in on this myosin head. So a much better depiction than what I can draw. So here's two of these molecules kind of wound together, and here's the myosin heads. And here's what a thick filament of mini myosin molecules held together look like. Okay, so that's the myosin molecule. And at the bottom of the page here is the actin molecule. Okay, so you've got these actin molecules that are strung together like two beads, two rows of beads, and you have these tropomyosin bands that wind around, and they're held attached by this troponin. So they're held attached to the actin molecules with this troponin. All right, so that's the basic structure within the muscle. And let's talk now about how exactly muscle contractions come about. So muscles contract via what is called a sliding filament mechanism. So a sliding filament mechanism. Okay, and the first thing that's going to happen is that a nerve impulse is transmitted down a nerve, so down a presynaptic nerve down to the muscle, which is going to be the postsynaptic tissues. So when this nerve impulse is transmitted down, it causes the release of a neurotransmitter. And in the case of muscle contractions, this neurotransmitter is acetylcholine. Okay? From there, we have chemically gated ion channels that open. And when these chemically gated ion channels open, due to the release and the binding of acetylcholine, sodium floods into the receiving membrane, so the muscle membrane. Okay? When this sodium floods into the muscle, the membrane depolarizes. Membrane depolarization causes calcium to be released from an area on the muscle called the sarcoplasmic reticulum. Okay. When the calcium goes throughout the muscle, it binds to troponin, which is on the actin filament. When that occurs, it causes a conformational change that pulls the tropomyosin away from the binding sites on the actin. And when this occurs, the myosin molecule will bind to the actin filament. So myosin filament binds to actin. And when this occurs, it automatically sets into a chain of reactions where those golf club head structures are attaching to the actin binding sites and basically ratcheting up the whole actin molecule. And this ratcheting, or this cross bridge, binding causes the sarcomere to shorten and that equals a muscle contraction. Eventually you want the myosin heads to release from the actin, and that is caused by ATP. So ATP is what causes the release of the myosin heads from the actin binding sites. All right, and so now that we've laid out these steps of this sliding filament mechanism of muscle contraction, let's look at a couple of the examples in the book and walk through what this looks like. So the first steps are that you get this nervous impulse. So the nerve impulse comes down from this presynaptic neuron. These synaptic vesicles release the neurotransmitter, in this case acetylcholine. Acetylcholine binds to receptors on this postsynaptic membrane, which causes the sodium channels to open. The sodium floods in. That causes the calcium to be released from the sarcoplasmic reticulum. Okay, so from here, let's go back to this. Once the calcium is released, the calcium comes in, binds to the troponin. The troponin undergoes a conformational change which pulls away these filaments of tropomyosin, exposing the binding sites for the myosin heads. So the myosin heads attach, automatically go into this cross-bridge response, or this cross-bridge cycle that causes the myosin to ratchet up that actin filament. And when that occurs, it looks like this. So you have a relaxed muscle here with these sarcomeres, and these sarcomeres are made up of actin and myosin filaments. And when the muscle is relaxed, it looks like this, but when it's contracted, these sarcomeres have pulled together. Okay, so you get a pulling together of those sarcomeres as the myosin filament ratchets up the actin. Okay, and then to get a release, we go through this cycle where you've got the myosin head that's bound to the actin, and here's the cross bridges that have formed, and you get ATP coming in to release this connection here. So to release this binding of the myosin heads to the actin molecule. If there's, at the same time as these are going on, you have pumps on the sarcoplasmic reticulum that start pumping calcium ions back into the sarcoplasmic reticulum, lowering the concentrations of calcium within that muscle. At lower concentrations of calcium, the troponin is released and relaxes back or undergoes that conformational change, which again covers the myosin binding sites on the actin so that the whole muscle can relax. Okay, and with that, that's going to end this particular segment. And thank you very much for watching, and we'll see you next time.